Welcome, everyone. Thank you so much for being here, and welcome to those of you who are watching online. 10 years ago last week, Nature published a paper that announced and described the sequencing of the human genome. This was already, at the time of the publication of this scientific paper, a much heralded achievement of the Human Genome Project. President Clinton had announced this success in a White House press conference the preceding June, well before the actual scientific publication. He called it at that time the most important, most wondrous map ever produced by humankind. This announcement was greeted with extremely high expectations that the discovery would change our understanding of human biology, of medicine as both science and practice, and of how we see ourselves as human beings. To what extent, we want to ask today, have these predictions proven true? Has the sequencing of the human genome been transformative? And in what ways? In science and medicine and beyond. In commerce and business, in regulation and law, even in ethics and philosophy. How are we all, and the kind of work we do, and the kind of lives we lead, different than they otherwise would have been? And what will the coming decades bring in this realm? There could be no better place to seek answers to these questions than here at Harvard and here in the company of my distinguished colleagues here today, who have generously agreed to assist us in this very broad-ranging effort to understand the meaning and the impact of this discovery 10 years out. I'll begin with questions to each panelist, engage them then a little bit with one another, and then take your questions. If you would be so kind as to write down questions on the cards that have been provided to you and hand them to the ushers, they will be brought to me so that I can then pose at least some of them to the panelists. Online viewers can pose their questions by emailing to the address that I think is going to be on the screen. It's questions@Harvard. Is that correct? questions@harvard.edu. So let me briefly introduce the members of the panel, not with lengthy introductions that tell you everything they've done. That would take the entire time allotted to us, but just a very brief effort at identification. Eric Lander is the founding director and president of the Broad Institute of Harvard and MIT, and he holds professorships in both Harvard Medical School and at MIT. Vamsi Mutha is associate professor of medicine at the Department of Systems Biology at Harvard Medical School and a senior associate member of the Broad Institute. Susan Lindy is professor and chair of the Department of History and Sociology of Science at the University of Pennsylvania. Vicky Sato is professor of the practice of molecular and cellular biology in Harvard's Faculty of Arts and Sciences and professor of management practice at Harvard Business School. She was president of Vertex Pharmaceuticals from 2000 to 2005. Margaret Hamburg is the Commissioner of Food and Drugs in the United States Department of Health and Human Services. She served as a Harvard overseer from 1999 to 2005 and is a Harvard alum. So let me join the panelists and begin the interchange. So Eric,
Drew.
I want to begin with you and ask if you perhaps could just give us a few comments on what the major expectations were, if you can remember back 10 years, for what the sequencing of the genome would lead to, and where we are now in relationship to those expectations. And I'd love to hear especially if there are any particular things that really have surprised you.
Okay, so expectations. Depends a lot on whose expectations you're talking about. My expectations This has gone so much faster than I had ever imagined. The work of sequencing the human genome involved an entire international effort to get 3 billion bases of DNA covered. Since then, just in those 10 years, the cost of sequencing has fallen by 100,000-fold, soon to be a million-fold, vastly faster than Moore's law in semiconductor, faster than anything that I've ever heard of. So it's meant that we can do today a billion bases, not in a year, but a billion bases at the Broad Institute in 5 minutes. So that has accelerated things unimaginably. But more importantly are the scientific discoveries that were unexpected that came out of it. Comparing genomes. As soon as you got the human done, we wanted the mouse and the dog and the rat and the aardvark and other things. And comparing all that, we've been able to read out the lab notebooks of evolution, so to speak, all the notes of the last 100 million years of evolution, and all sorts of surprises have emerged. There are a lot fewer genes than we thought, but an awful lot more regulatory controls. It's remaking our picture of the biology of how you run human or other cells. Disease. If you go back 10 years ago, the number of genes that we knew that were associated with common diseases, I don't mean rare Mendelian disease, but common diseases, diabetes or Alzheimer's or psychiatric diseases, was about 20. Today, it's about 1,100. And the knowledge that's emerging about diseases, 100 genes related to inflammatory bowel disease. The first set of genes, about a dozen now, related to psychiatric disease, pointing to for the first time to the kind of pathways that are involved in those diseases, giving us our first clues. Here at Harvard, actually I'm picking things that are all at Harvard, in fact. Here at Harvard, an amazing study that's shown why African Americans have a higher risk of kidney disease. And it traces back to an African allele that looks like it was protective against sleeping sickness, but has this liability. So we've got about 1,100 examples so far of genes that connect and point to common disease. Cancer, same sort of thing is going on. There's an explosion of work that will culminate, I think, in the next 5 years in a pretty comprehensive list of all the targets that get mutated in different kind of cancers and give us the kind of roadmap for finding the Achilles heels of cancer for therapeutics and diagnostics. Human history. That's been, I mean, you're a historian. There's some pretty great history in the human genome. You know, for the first time, we can read out stories of positive selection, changes that happened in the genome that were advantageous. The gene that gave people in Northern Europe and in East Africa the ability to drink milk as adults, for example. We can read out stories not just of how the world was peopled by humans spreading out and dividing up into different groups, but mixtures upon mixtures. And the coolest mixture, again with a Harvard connection, is the recognition that we mixed with Neanderthals on the way out from Africa, and about a quarter of your genome is Neanderthal, for example. So all of that is quite amazing. Now, you know, the single most wonderful thing about all of this is what it means for young students today, what it means for undergraduates and graduates and postdocs at Harvard and MIT is they have the tools to do things today that used to take armies to do. So it's unleashed an amazing creativity. The Genome Project was thought of 20 years ago as big science, and maybe it was big in some ways, but it's ended up being the enabler of small science in a really dramatic way by putting information and tools in the hands of everybody. So my expectations have been blown away. But then you have to ask, what about public expectations? When the human genome sequence was announced, and it was announced multiple times, press conferences and then the paper and all this, there were a lot of expectations set that were fabulously naive. Journalists wrote about how we were going to now have drugs for all these diseases in the next decade, and we were going to understand everything and cure everything. And somebody was smoking something. This is just nuts. You know, that is not the way that scientific progress proceeds. We got the first read of the genetic code. The first thing we've got to do is, well, we got to fill in all the gaps in it. We then had to begin to figure out how it varies between people, look at it in diseases. We're at the point now, a decade later, where we have all of this beginning knowledge about pathways. But as Peggy will surely tell us, just the path from the early part of a discovery process for a drug to the clinical trials, to the approval, to reaching patients, that can consume a decade. Real scientific revolutions are measured over the period of multiple decades. It was 60 years from the discovery of the germ theory of disease, that microbes cause disease, to when we had cheap antibiotics available for the population. I think it'll be somewhat less than that, but we all do this for our children and our children's children. Anybody who's selling you the line that this is going to have an impact by, the third quarter of the year for stock price purposes, or even within, the next election or two, that's not the time frame at which revolutions happen.
Thank you. We just all are looking for immortality, Eric. That's why we're impatient.
Have a good time while you're here instead.
Yeah, that's a good advice. That must be the philosophy part of this discussion, right?
I think so.
Vamsi, you began your research career just as this sequencing effort was beginning to bear fruit. So what is your experience as a kind of early adopter of this sequence and as a young scientist in the field? Eric said a little bit about what this has meant for students and young scientists, and perhaps that's been part of your experience.
Sure. So I think almost exactly 10 years ago, I was finishing my clinical training at Boston's Brigham and Women's Hospital, one of the Harvard hospitals here. And I had spent many sleepless nights during that time, in part because of the grueling call schedule, but also because I wasn't exactly sure what I was going to do after my clinical training. Prior to that, I had trained in mathematics and computer science at Stanford. And during my medical school and my clinical training, I developed what would end up being, I guess, a really deep and enduring passion for mitochondrial biology. So these are the structures within our cells that are responsible for making our energy currency. And I became fascinated by these mitochondrial disorders that are classified as orphan diseases. These are rare, devastating disorders. We didn't know the molecular basis for the majority of them, but I was absolutely fascinated by them, and in part because they were orphan diseases, I was more interested in them. And so I had these sleepless nights trying to figure out what I was going to do after my clinical training, and the two things that I did know was that I loved mitochondria and I loved math. And so here in Boston, in the biomedical arena in the late 1990s, early 2000s, there was almost a palpable enthusiasm about genomics. The human genome sequence was going to be coming online, and I could feel that energy. And one of the statistics that really gripped me at that time was that there was going to be about 20,000 human proteins. The number bounced around. But all of the previous literature, all the biomedical literature, all the previously known drug targets, all of the previously known disease genes neatly mapped onto at most 5,000 of those proteins. And so what that meant, I could do the math. There's 15,000 proteins out there about which absolutely nothing was known. And as a relatively young scientist at that time, I felt that I could make a mark and basically study some new proteins that no one had ever studied before. And that's what drew me in, the opportunity to do something really, really new. And if in the process I could use a little bit of math, that would be fantastic. And so over the last 10 years, first as a fellow and then as a professor in genomics, we've been trying to go after these mitochondrial disorders that had grabbed me in medical school and in residency. And 10 years ago, there was about 10 genes that were known to underlie, 10 nuclear genes that were known to underlie these disorders. But that only accounted for a very, very small fraction of all of these disorders. And in the same way that you can't diagnose a problem with your engine if you don't know all of its components, I also reasoned that we needed to know all of the components of this organelle in order to diagnose the rest of these cases. And so I started at that time using methods that were largely inspired by the Genome Project to systematically identify all of those components that make up this cellular machinery. And over many, many years, we've now identified the 1,100 components of this structure. And now what we do is we do a typical genomic experiment for us is to take those 1,100 genes, we cluster them, we have matrices that we can generate using freely downloadable data. We diagonalize these matrices, we cluster them, and we look for patterns that might hint that a particular gene might underlie one of these disorders. And over the last 10 years or so, we've identified about 10 more of the rare Mendelian diseases. And even though they don't impact most of us in the audience or friends that we have, they really have had a real clinical impact. Genetic tests are available for most of these right now. And a few years ago, something that really moved me was I received a Christmas card from a couple in Canada that had lost 2 kids from one of these disorders. And they had utilized the genetic test, the gene that we had discovered. They utilized a genetic test that allowed them to ensure that their future offspring would not have that particular mutation. And that Christmas card included a birth announcement of a little baby boy that's now hanging in our laboratory, and it serves as a reminder of why we do the work that we do. And so that, when I left sort of the clinical world and started focusing on research, I'd always wondered whether the work I did would actually have any real relevance. And I knew that we always had a tangible relevance, but this was more than tangible. So over the last few years, something new is happening. Something really exciting is happening right now, and that's this era of individual genome sequencing. And this is what Eric alluded to. And this is being enabled by this new technology that we call next-generation sequencing. And this is extremely exciting. And this is a technology that a number of teams around town elsewhere are going to be using in their campaigns against diabetes and cancer and a neuropsychiatric disease. And so even though the narrative that I shared with you right now is focused on mitochondrial disease, what's really exciting right now is that the exact same thing is happening, even on larger scales, against the common diseases. And that's what makes this sort of era of genomic medicine so exciting right now. And so I'm really excited about the future, and if I were, a college student or a graduate student right now, I couldn't think of a better time to be embarking upon sort of biomedical research than right now. But there are a number of daunting challenges that lie ahead, and I think we're going to need the help of the young students over here in order to be able to solve those problems. And if I have a few more minutes, I'll just highlight some of these challenges, if that's okay. These new sequencing technologies are just so high throughput. They're generating so much data. We're facing what a lot of people are calling a genomic tsunami. There's thousands of genome sequences that are literally being sequenced right now and being put in the public domain, but we're actually having difficulty even storing all the data. And most of our computers can't even upload these data sets into memory without crashing. And so it's almost as though Moore's law is unable to keep up with genomic sequencing. But I think the greatest challenge moving forward actually lies in interpreting this DNA sequence information in a way that's clinically useful and also responsible. So we're entering upon this new era of genomic medicine, and it's going to require retooling on the part of our students, on the part of our scientists, and on the part of our physicians. And the skills that are going to be needed to succeed in the future are probability theory and databases. And I think medical education has to change dramatically as well. So I envision a future in which the Harvard Medical School students, for example, they're going to be taking their class on the introduction to the physical exam, but perhaps they're going to have another class called an introduction to the genome exam. And they're going to have to learn how to basically zoom in and out of the human genome sequence, perhaps with a web-based portal that allows them to simultaneously evaluate the DNA sequence while they're also evaluating bumps and scratches on the body. And this has to be done iteratively in a seamless manner. That's the only way to interpret the genome sequence information. We're going to need some really smart biologists that are going to understand the grammar of the genome. And it's only if we understand the grammar of the genome that we'll be able to understand that remaining 99% of the human genome that Eric and others speak about. And we're going to need some really smart entrepreneurs that are going to commercialize all these new discoveries so that we actually have drugs that we can give back to the patients. And we're going to need policymakers that are going to be able to be well-versed in genomics so that we can make sure that this information is used in a fair way. So I think the last thing I want to just mention is that I think the one key lesson that genomics has taught us is that there's real value to teamwork. None of the work we've done, none of the work that others are doing in this area is done by an individual. It takes teams. And this is a new way of doing science. And I think at Harvard, we really have a competitive advantage thanks to the community that includes our medical school, our hospitals, the Broad Institute, neighboring institutions. So we really have a special opportunity over here. And I guess my sincere hope is that some of the students here today, you'll be willing to join some of these teams, hopefully even lead some of these teams to help usher in this new era of genomic medicine.
Thank you, Vamsi. This era of genomic medicine, Susan, is something that you've thought a good bit about, but from a bit of a different perspective. What are the challenges this poses for patients and families and the recipients of the benefit of these discoveries, or the difficult questions posed by the insight and information that these discoveries impart?
When we originally talked about this this panel, there was a sort of claim at the beginning of our discussion that the Genome Project had had no real impact on healthcare, and I objected that it has indeed had a massive impact, but a mixed one. And so I'm going to talk a little bit about what I call ambiguous precision. And when Eric was talking about the escalating rate of information and discovery, I would suggest there's also an escalating rate of ambiguity and an escalating rate of what I sometimes call uninformative information. And that individuals, that is, consumers, patients, families, people considering reproduction, are in the present and leaving aside Vamsi's expectations of what the future might be. What we have on the ground right now is a very complicated operating, I could call it a machine or a system, that's generating a great deal of information that people, the information that it is very difficult for individuals to assess and to interpret. So I'm just going to I've worked on the history. I have a very different kind of expertise. I'm a historian of science, of biomedical science. I've been working on the history of a particular disease, a very interesting, an orphan disease, which Vamsi probably knows, which is cystic fibrosis. And what's happened with cystic fibrosis is that over the last 5 to 7 years, it's been very rapidly added through consensus agreements and through state panels to one of the diseases for which reproductive genetic counseling is recommended. The gene for CF was discovered in 1989 by Francis Collins. It is a canonical and very important disease in the history of the Human Genome Project. It's also important because it was the first disease for which rapid and effective gene therapy was expected, promoted, heavily financed, and then did not ever happen. In one of our interviews, I actually am working with a CF patient who's been helping me with this project, and in one of our interviews, she encountered a researcher who said he considered not studying CF because he believed in 1992, because he believed that soon the disease would be gone. And so it was expected to disappear so rapidly. Cystic fibrosis has not disappeared. It's a disorder that leads to excess mucus production. It has pancreatic effects, and it is caused by a mutation, or by many mutations actually, in the CFTR gene. And mutations in CFTR have been, at least 1,500 different mutations have been identified associated with the actual clinical syndrome of CF in this particular gene. So what happened is that newborn screening for CF began very early in the 1990s, and many state panels, as many of you probably know, they add additional diseases to these newborn screening panels. They began in the 1990s, and particularly for one of the most important mutations, which is delta F508. After 2002, when reproductive genetic screening became the norm in many different states and in other places around the world, partly because of a consensus on the part of geneticists and clinicians that this was an appropriate thing to do, the rate of infants born with delta F508 dropped 50%. So in other words, when you tell people who are considering having children that they are carriers of delta F508, they do something that leads to a statistically significant, to say the least, epidemiological effect that's very fast. So in California, the fall was only in a single year after the introduction of reproductive genetic counseling, infants born with delta F508 dropped 50%. That's pretty dramatic. And you might want to say, well, cystic fibrosis is a serious genetic disease, and therefore we can expect people to make health decisions along these lines. But in fact, two people who both have delta F508 can have a dramatically different clinical outcome. The arc of their lives is not predictable. Their clinical experience, their health, their everyday functionality cannot be predicted by the presence of that particular mutation. And in addition, there's been some wonderful work on the extreme variation between CF centers and clinics that are using exactly variations in survival and overall health in clinics that are using the exact same CFF, Cystic Fibrosis Foundation protocols, which are quite elaborate and strict and very involved. So what I want to suggest is that in the genomic age, what is happening already and what is likely to escalate is not only our information about the genome, but the kinds of conflicted information that individual consumers have to process, have to deal with. They have to make individual decisions in a climate that is not nearly as solid and reliable as some accounts might make it seem. And so it's a kind of lonely choice. It's a choice with profound social consequences. And as more and more diseases are added to these panels as appropriate for reproductive genetic counseling, the overall health consequences could be profound in many different ways. So my closing comment is just this, and I know everybody here on the panel would agree with me. Genomics or genes are not exactly the same as health. And if genes are complex, intricate, baroque, even bizarre, and Eric has beautifully captured that in some of his work, so is health. And health is just as complex, just as baroque, just as bizarre, and it takes just as many tools to figure out how you get somebody to that state.
Thank you, Susan. And part of how you get someone to that state is the realm of life in which Vicki has been involved. So how do you see these past 10 years, and how do you see the translation of these discoveries into the realm of health, taking into account both the science that we've been hearing about and the kinds of questions that Susan has raised?
Well, you know, inventing medicines is a very practical science. And the discipline of inventing medicines, as Eric alluded to, is one that takes a very long time because our knowledge of human biology and human diversity is so imperfect. And that lack of knowledge, that the imperfection of our knowledge there has really led to an industry that is by its nature empiric, by its nature inclined to fail because our knowledge base is low. And I think the promise of the genome brought with it a tremendous public excitement about the transformation that was going to occur or could occur in the pharmaceutical business as a consequence, part of the hype that didn't make the job any easier. And I think all of us on some level, those, you know, as patients, as scientists, as drug discoverers, we were beset with this image, if you will, the 20th century image of Paul Ehrlich's magic bullet that went, aha, we'll complete the genome, and the next day we'll say, there's the gene for cancer, and there's the gene for schizophrenia, and there's the gene for type 2 diabetes, and then the drug companies could finally get to work, and we'd have profoundly better medicines. At least, and I think we still carry around that image. And as Eric alluded to, 10 years is about half the time it takes to bring a drug from an idea that Vamsi might have to a medicine that Peggy might have to approve. So 10 years isn't very long. But having said that, I think there is no mistaking the fact that genomic sciences, the completion of the human genome and its penumbra of other discoveries, has changed the way we think about making medicines. And 10 years is not very long. So I sort of think about this as like making and testing a drug. It's a three-phase issue. In phase one, and I remember saying this about 20 years ago when people were postulating the genome, I said, the first thing that's going to happen is that drug discovery is actually going to get slower. And I think it did for a while because, to a point that you made, Vamsi, there was so much information of a type that drug companies, biotech companies were unprepared to digest, that we were the bottom of the tsunami. The infrastructure for making drugs wasn't designed to match the rate or the kind of information that was coming from the human genome. And even scientists can be slow to change how they solve problems. I mean, Arthur Kornberg, a Nobel laureate, once said that scientists have a tendency to fondle their problems. And I think that's true. We have a tendency to fondle the way we do things as well as our problems. And it took the industry, despite its excitement, I think the first phase of several years to really change the way it worked to take advantage of genomic information in a way that could start to translate into improved insights into medicine. In phase two, I think we've seen that the infrastructure has changed, our vocabulary has changed. We're using the language of genomics and genetics to describe diseases that we didn't describe in those words before. We don't talk about cancer anymore as uncontrolled growth, which is a pretty scientific definition in its way, but we talk about every different cancer in the molecular language of genetic mutations that lead to that particular cancer. I think we've come to the realization, which is a daunting one, that every cancer is unique and that every cancer has a natural history that causes it to be different over the years over which one carries the disease. Our understanding of inflammatory diseases has changed profoundly. We use a different vocabulary because our understanding of diseases like arthritis and lupus is defined now by new information from the genome. So our understanding of disease has been profound. The pharmaceutical industry has also benefited from the genomics of model organisms. It's benefited from two things, in addition to the human genome. One is the technology that's been alluded to. The ability to do rapid sequencing allows us to evaluate patients in new ways. It allows us to select patients that might be better suited for particular medicines. It allows us to understand that there's sufficient molecular variation that medicines that work very well for you in your disease might not work very well for you, even if you have the same disease. So that technology of rapid DNA sequencing is allowing us to match our medicines to patients much more effectively. It's a different concept. We were very used to in the pharmaceutical business of saying, Claritin, it's a non-sedating antihistamine. It only works in about 40% of the people, but we really don't understand which 40% it is, so let's all take it. The ability to understand the entire genome of test organisms, rodents, dogs, microorganisms, has allowed us to use those test animals much more intelligently to both understand disease and also to evaluate our molecules. So in addition to the human genome, the completion of the genomes of many test organisms has allowed us to link drug action to disease in ways that we can't yet do in humans, actually. So I think that was another part of phase two. Phase three now, we're seeing the first products of not genomically driven so much as genomically informed medicines and diagnostics coming to the market. So we're seeing diagnostic tests that allow us to determine, again, many of the advances have come in cancer first with the work in the cancer genome as well. But we can say, if you carry a particular mutation, this particular drug really isn't going to work for you, even though if you have the same kind of cancer, colorectal cancer, and you don't carry that mutation, this drug could actually be a remarkable drug. So the ability to use genetic and genomic information to differentiate between people who will respond well and people who will not respond is already affecting treatment and raising some very interesting issues about treatment dynamics. Because of course, this science isn't absolute. We know that in general, and actually with a fairly high degree of precision, that it's highly likely that if you carry this mutation, this drug will not work. But in a disease where the consequences are as dire, it's very difficult to say, do we know with 100% accuracy that if we withhold that medication, that it wouldn't have done some good? I think that was grammatically incorrect, but you get the drift. We're seeing new diagnostic tools that are starting to tell us, for example, in prostate cancer, whether you have a form of prostate cancer that is indolent and may really be best managed with a watchful waiting over the next 20 years, or whether you have the kind of prostate cancer that's aggressive and really should confront some of the difficult choices that surgical radiation therapies, surgical and radiation therapies might afford. So the impact of this kind of information and how we treat patients today, only a decade later, I think is remarkable. It has been an accelerant for informed treatment. It's been an accelerant for informed treatment. It will continue to be an accelerant for informed diagnosis, hopefully linked to better treatments. It's interesting that you brought up CF. One of the last projects that I started to work on when I was at Vertex actually was with the CF Foundation, where we decided to take on the challenge of seeing if we could make a medicine that actually struck at the heart of the genetic alteration in cystic fibrosis, the CFTR gene that Susan alluded to. Most of the other treatments for cystic fibrosis treat the symptoms of the disease and have been quite successful in prolonging life, actually. But these would be the first medicines that actually attempted to correct the genetic defect. And 2 of those drugs are in clinical trials now, and those 2 different drugs actually address probably 2 different mutations that contribute to the panoply of cystic fibrosis. So for that to be true, and I think Vertex is hoping that one of those drugs might actually make it to the FDA by the end of 2011, to go from, and I have to say, in the 20 years preceding that, from the discovery of CFTR to the time Vertex started that project, nothing much happened. So from, when did we start? Probably 2000, 2002, to move that quickly based on new insights and tools from genetics and genomics is, I think, a reflection of the fact that this kind of information is not only powerful, but it can be acted upon with some speed. So I'll add my voice to the fact that one of the reasons I came back to Harvard after spending 25 fantastic years actually learning about and making some medicines is because now is the time to be a scientist that makes medicines, because the tools that are available, driven by the technology around genomics really, allows us to move from saying, hey, there's some mold on this bread, what are the odds that it actually kills bacteria, to saying, we can actually move from molecular insight to molecular solution with a kind of direction and precision that I could only dream about when I was an undergraduate here. I went back and looked recently at the textbook that I used in cell biology, and things that we thought were truths, right? We thought, actually the new truth was DNA made RNA made protein, and that was all you need to know on Earth. So much has changed in a relatively short period of time, although I am old, that I cannot imagine what the medicines of the future might look like. We all carry around this idea of not just the magic bullet anymore, but of my magic bullet and your magic bullet and your magic bullet. I don't know. I don't know. I'm excited about the possibilities. I'm excited to think that diseases that we don't even understand yet will be treated in a decade. Do we understand schizophrenia? I don't think so. Can we see a path to treating it? Can we imagine such a path? We can imagine that. Unimaginable 5 years ago. Unimaginable. So it's time to pick up a pipette.
So Peggy, the medicines of the future, they rest in your hands in some ways in terms of evaluating their appropriateness and their safety. And what are the public policy implications of all that we've heard? And how do you think about the proper way to advance the public interest and to defend the public interest in face of these important new advances?
Well, first I just have to make the comment that it's a whole lot harder to be a student today, I think, than back when I was in medical school and it was just learning DNA makes RNA makes proteins. So I'm sort of glad, even though FDA commissioner is not an easy job, it may be easier than being a medical student today. But in terms of where we're at, I think it's clearly the case that the implications of the sequencing of the human genome are enormous, but that we're in the very early stages, and that just as the science is in the early stages of development, the truth is that the regulatory process is also in an early stage of development as well, and it's very, very important for these two overlapping communities to work closely together as discoveries unfold and opportunities present themselves. I think it's clear that while the genomics arena is still very young, that it is permeating biomedical research in very profound ways with huge impacts. It is also clearly shaping the pipeline to deliver the real-world products of the so-called genomic revolution, that we're beginning to be able to trace the development of basic discoveries into candidate interventions, followed by further research, including clinical studies, through to the regulatory review and approval process and into clinical practice and medical care. While we, I think, have long understood that this understanding of the genome was going to be transformational, I think what we really didn't appreciate was how difficult and complex it was going to be to really convert that science into applied solutions for patients and consumers who need them. And that the successful development and integration of so much new knowledge and capabilities was going to require both continuing work on the part of the basic and clinical scientists, some of whom are represented here on this panel, but also that we would need to think about the successful integration into medicine and the strategies to prevent disease and to promote health, and all of what that would require. Again, we've heard some of it on this panel in terms of the education of healthcare providers and the public. We haven't talked about some of the issues around genetic discrimination and the need to really protect privacy in some key areas. And we also haven't talked about the oversight system that ensures the accuracy and reliability of genetic tests and the application of genetic information to permit safer and more efficacious therapies or use of drugs. And that's clearly what I'm here to really focus on. And this is absolutely key because the FDA needs to have a strong, meaningful, and clear presence. We recognize that we have a pressing responsibility to help speed the translation of new insights, opportunities, and research into solutions for patients and consumers, but that we are, too, traversing new ground. Clearly, this is a critical moment as we're really beginning to integrate genomics into medical care, and I want to just point to a few key areas. There are clear advances in existing applications. Pharmacogenomic information, that is to say the information about how variations in the human genome affect the response to medications, is increasingly available to healthcare providers, but it is still limited. Some 70 FDA-approved drugs now contain pharmacogenomic information or genetic testing information on their labels. And this type of information can help guide the treatment and management of patients and enhance safety and efficacy in terms of drug use. Some of the earliest applications have been in the area of appropriate drug dosing. The drug warfarin, which is a blood-thinning drug, is one good example. It's notoriously hard to get your patient on the right dose of warfarin. And in fact, one of the leading causes of ER visits among the elderly is complications of warfarin therapy. But the FDA, working with other scientists, was able to validate a genetic test that helps healthcare providers estimate what is the right, an appropriate dose of warfarin for their patients. And that's now part of the label, and it provides healthcare providers with a new tool to offer better, safer care. Moving forward, we'll increasingly be focused on the opportunities and complexities of targeted therapies for individual patients, so-called personalized medicine. And we can use the insights of the genome to match an individual's genetic profile to the likely effects and potential benefits of a drug treatment, as you've heard. And this is already happening in a couple of key domains. It was mentioned about the ability to identify who will or who won't respond to a given therapy based on specific mutations that they may have in a cancer. For example, the colorectal cancer and the KRAS mutation that was just mentioned by Vicki. We can also identify people who are prone to toxicities. For example, the hypersensitivity reactions that are seen in carriers of the HLA-B5701 allele and the use of the HIV drug abacavir. And this kind of work that really identifies a genetic marker and a drug treatment response relationship is key, I think, to the future of the full utilization of the insights of genomics, but it will require new ways of doing research and new relationships between regulatory science and the rest of the scientific enterprise. It will require new and innovative clinical trial designs so that we can stratify populations and study the appropriate subgroups. If not, many of these important treatment differences will get lost. They'll get basically averaged out. And this work clearly needs to be done, and it needs to be done with the combined efforts and expertise of academia, industry, and government. We at the FDA also need to work to develop clear regulatory frameworks and pathways for drug diagnostic test combinations that support innovation, that foster and facilitate the kind of clinical investigation and treatment opportunities that we've just been talking about, and that fuel the drive towards stratified or personalized medicine. And of course, findings of genetic research can also be used to identify promising drug targets for a range of disease conditions, and Vicki also was talking about that. Oncology is a prime example where genetic testing can type tumors and help define pathways of transformation for targeted drug development. Cystic fibrosis is one great example. We're also seeing in the oncology area many new strategies and a lot of promise in terms of this kind of targeted drug development approach. But developing the right genetic tests linked to treatment is key, and we need to ensure that these tests are analytically and clinically valid, that they detect the right biological target, and that the proper use of those tests in the clinical setting and the indications for the resulting treatment and medical interventions are fully validated. And of course, there are many challenges in diagnostic development. And for better or for worse, we expect that the validation and qualification of novel biomarkers using genetic tests will only become increasingly complex as we move to large panels of SNPs, or more significantly, as we move to whole genome sequencing. So FDA must work together with industry and with the academic community to define how to determine validity. And when we have data, we must ensure that its clinical utility is established and clearly defined, again, working with partners in industry and academia. Moreover, physicians and the public must be on the same page as we learn to interpret clinical meaning in sequence information, and we must all work together to determine how to best use available and growing future databases that include genetic information with medical records to gain new understandings about the nature of diseases and how to treat and prevent them. So at the end of the day, there really is just an enormous amount that we're focused on. The regulatory capacity and procedures must be fundamentally linked to the way that the science is being done, to the way that the drug development is being targeted, and to the way that these new medical products will be used in clinical practice. I guess I should conclude, and maybe the best way is to use an old academic refrain that more research needs to be done, but in the meantime, we have a lot of work to be done, and I think we really need to do it together. And I want to emphasize what Vamsi said about the importance of teamwork. I think that perhaps more than in any other area of medicine or public health today, success will require partnership across disciplines, across sectors, and importantly, amongst scientists, healthcare providers, regulators, and consumers. Thank you.
Thank you, Peggy. I'm struck listening to you to all of you, that it makes much more sense to talk about genomic revolutions, plural, than a single revolution, because you've talked about a variety of ways in which the last decade has been transformative. Vicki, you talked about different vocabularies. And Susan, in a sense, you spoke about a different role for the patient. And both Eric and Vamsi touched on the word team and the nature of that, and you used it in a slightly different way from the way I want to focus on it for just a second, which is, how do we do science now in this era? And isn't part of this revolution in the way you pose questions, in the way science is organized? And I wonder, Eric, you've written a little bit about that. Vamsi, you're living it and thinking about it. If one or another of you or any of you would say a word about that part of this revolution.
I'll say just a little bit, which is every science at some point goes through the revolution where it sees its big picture, where you see the entire geography of the Earth as opposed to the little bits you live on, or the whole periodic table. It's some kind of intellectual enclosure act. You know it's finite, you can see the big picture, and you can start asking about patterns of the whole thing, why South America and Africa fit in with each other, or why the elements are in the columns of the periodic table. So there's a total shift when you start thinking that way, but it also means that what it takes to answer those questions may be very different. In genomics today, you can't possibly do it without computer science and mathematics, because it's all about information. But you also have to deal with, big experiments, so you're going to need friends who are engineers and chemists. And of course, the organism you really want to study is the human patient who's in the hospital. And so there's a need to come together in teams, and the usual model that works for many, many things of an individual faculty lab with a couple of students and a technician, which is still well suited to many things, may not be well suited to solving a lot of the problems we have to solve today. So the question is, how do you organize teams that are not hierarchical things where some are subordinate to others, but alliances of people who come together, and sometimes you're leading and sometimes I'm leading. And I don't think the academic world has coped with that so well. We tend to reward the first author on the paper and the last author. We tend to think the right unit is the lab. I think it's a fascinating stress on what it means to do science today.
Vamsi, do you have any
I'll just follow up a little bit. I think what's really exciting now is over the last couple of years, almost all of our papers, it includes a combination of a computational person, a wet lab biologist, and a physician. And the papers that we produce, are infinitely more exciting than the ones that I wrote as a single author some 15, 20 years ago. So it's just really exciting to have these types of papers. But there is this issue of how do you incentivize the individuals? And I've seen something transform over the last 10 years. And I'm reminded of something that one of my friends from Wall Street once told me. He said, we have a very simple way of incentivizing people, teams on Wall Street, because money is infinitely divisible. But unfortunately, authorship is not. And I remember about 10 years ago or so, there used to be a lot more authorship squabbles, but now something is changing. Now the team actually asks, what's the best project that we should do? What's the best science that we should do? And they have faith that the authorship will work itself out at the end. And we use practical strategies like a lot of stars for equal contribution. And so I do see a change over the last 10 years, which is really exciting.
And Peggy has emphasized the need for teams beyond simply the laboratory teams that stretch into the commercial world and into the regulatory world. Susan, you look like you wanted to make a comment.
Another thing that I see emerging at many institutions is an interest in bringing into genomics research, particularly persons who have either ethical or sociological or historical research. I'm affiliated at Penn with the Center for the Integration of Genetic Healthcare Technologies, which is an NIH-funded center which brings together scientists, clinicians, sociologists, other kinds of humanists, and there's a sort of conviction in the scientific community that persons who have other kinds of expertise should be a part of moving forward appropriately with these kinds of information technology.
So many of the questions I've been given are about ethics and about what are the implications. Let me just read one of these, and I could have picked any of it, about 8 here. What key moral dilemmas do you foresee as we move into the next generation of sequencing, and particularly into the commercialization of genomics? That leaves it nice and broad. Any of you want to jump in on any of those?
I want Eric to answer that one. I know what I think. So.
Tell us what you think, and then I'll tell you what I think.
Well, I've already told you. I think the big concern, I've begun to think of genomics as an information problem. If you think about it, even when Jim Watson published his own genome, he left a gene out. You guys know it's APOE. He didn't publish that one, and he didn't want to know whether he had it or not because it's associated with a heightened risk for Alzheimer's. So he himself is making a decision to let everybody know everything about his genome except this one thing. I'm a participant in a human subjects research at Coriell in which you give your entire genome to Coriell. They can study it, they can find any genes they want, but they're not going to tell you about anything that you can't have any impact on. So they'll tell you about genes that might increase your risk for heart disease, but they're not going to tell you about APOE or other conditions. And so, and also there's NPEs, non-paternity events. It's common in contemporary cytogenetics labs when you get someone in for, say, a microarray to discover that the father in the family is not I don't know how, I'm not sure about the percentage, but it's surprisingly common. And so what happens is that the people in the laboratory know something relevant, we might say, to the family, and they do not tell the family. So rules about information management, who knows what when, who is who can hold information, who has a right to it, and what kinds of information should be withheld or protected. I think, and it's very complicated.
And you know that Jim Watson did not succeed by withholding that.
Oh, what happened?
Oh, well, it turns out that the nearby genetic markers are correlated enough that you can infer his APOE genotype from the nearby markers and knowledge of the human population. So it tells you just how complicated it is. You may think you're blacking that out, but it doesn't work.
Yeah.
So, Eric, what's your take on this one, the commercialization and the ethical dilemmas?
I worry about a couple of things. One is equity. As we increase the possibilities for doing so many things, we just deepen and deepen the equity issues about who has access to those things. That worries me a great deal. The other is maybe, as much as I'm a genomicist, I'm going to say genomics doesn't explain everything. There's a real risk that we are going to forget that there are other really important solutions. You want to do something about cancer? Stop smoking. You want to do something about diabetes? In many cases, not all, there are effective therapies, exercise and diet. And we haven't figured out how to apply those. We haven't figured out how to create the right incentives for people to use those. So the genome is an incredibly important foundation for all sorts of research. It will be transforming and it'll solve certain problems that can't be solved any other way. But thinking that the world is about getting the balances right, one of the things I worry about is whether we will in fact be distracted from solutions that we know should work. So it's basically seeing the genome in context.
And Vicki, how about your take on this?
I was actually going to move in a slightly different direction on this issue, because to your point, Eric, that there is no question about the impact of genomics and of the human genome on many of the things we're talking about. It's changing the face of how we think about disease, and it's changing how we think about what to do about disease. But there are a lot of problems that genomics isn't going to address. And I think the other is this issue of teamwork, is that genomics has bred, and this is really off the topic, we can talk about ethics later as well, but that the teamwork that was involved in solving, in addressing, completing the human genome, has led to a model for research that is, as you both described, perhaps ideal for solving problems. Right? So to the extent that we want to solve the problem of making a drug for a complex disease, or we want to solve the problem of understanding a complex disease, teamwork and the kind of multidisciplinary teamwork that we're talking about is both essential and fantastic to see. But one shouldn't, I think, then conclude that all science needs to be done in this manner.
Certainly not.
You know, I think profound insights about how the world works, how we work, are going to come from very different models of scientific exploration. And the beauty of the individual scientific explorer can't be abandoned here, I think. So a place where genomics might have done us a bit of a disservice is in overbalancing, if you will, some of our assumptions about how science has to be done. And I think you alluded to that yourself, Eric. And so I think that's an important issue about some of the collateral impact of genomic science.
If I could just make a slightly different comment also, I think that all of this discussion, I think, underscores another sort of ethical responsibility that we have, both as educators and as government entities and as citizens, which is to really strive to enhance education about science and enable people, both the general public and also, I think, healthcare providers, to be able to cope with this tsunami of information, to be able to tease out what's the uninformative information from the really important information, how to help people deal with what has always been, you know, a terribly difficult issue of risk communication and risk management. And then I think, you know, that is part and parcel of this enhancing the general level of understanding and education about this set of issues. I think, we have a real responsibility, especially as we move ideas into real-world products, to really, adhere very strongly to the notion that we have to assure the validity of the genetic tests, that they really are identifying what they purport to do, and really try to do the best possible job. It's very, very hard at linking the relative risk of a particular genetic marker with an actual risk for disease or opportunity for disease intervention. And I think that right now we play a little bit fast and loose in this area, and it may not serve the public well.
We've actually gone over our appointed time, but let me end with a question that came from the audience for any and all of you, which is about the Sputnik moment we have been challenged to rise to. And how do you size up the current debate on research development and the future of science in the United States, and how do we relate to that as we think about these very important scientific issues that before us?
Well, I think this is absolutely a Sputnik moment for biomedical science and research. This is clearly the century of biomedical science, and I think that as a nation, we really should mobilize to capture it. It involves, obviously, the contributions of genomics, but it is broader. But I think that when we think about what really matters to this nation today, from what matters to people and their health and the health of their families and communities, to strategies to provide the best possible health care and to also reduce costs, to what do we need to do to support our economy, to support high-value jobs, to support global economic competitiveness, the opportunities to really do some big and important things in the biomedical science and research arena are absolutely profound, and we should seize them.
I actually think it's more complicated than Sputnik.
Well, that may be.
Because I remember Sputnik, and I remember being the benefit of the fact that a tremendous amount of federal money went into K through 12 education to try and make sure we didn't lose the whatever race we were racing the Russians for. And that had to do with translating and creating things, and be they weapon systems or defense systems or whatever. The impact of biomedicine and these discoveries in biomedicine are not just about the kinds of products we're talking about, whether they're drugs or diagnostics or whatever. It actually puts a burden on all of us as individuals to understand enough about our health, about our biology, about our biochemistry, to make intelligent decisions about how we live, how we live for health, what medicines we take, how we take them. So it's not a matter of educating a population of people who will build a better rocket ship. It's about educating everyone, because everyone is going to have to take much more responsibility for their health care in the future. I mean, I think healthcare is going to become increasingly patient-driven. I think that's a good thing, but that means that every person has to actually learn some biology and has to learn some biochemistry. You can't skip that class and if you want to make an informed decision. So I actually think the educational consequences of this revolution, or revolutions, is much more daunting. And if we fail at it, the health consequences, the economic consequences, the competitive consequences will be significant. In some ways, more significant than losing the race for space.
I think
Personal
we won.
opinion,
We
right.
won the race for space.
Whatever, right?
I have a slightly different perspective, and it's pretty short, but it's just that it would be impossible to overestimate the impact of science, technology, and medicine, and generous funding of science, technology, and medicine on the American century, on the last 100 years, and the state of this country, and all that we've been able to accomplish. The support for science is central to economic security, military security, social life, and so it's money well spent, and it should be an absolute top priority for all the reasons you suggest, not just because of personal health, but because it's central to the state of the nation.
Absolutely.
Yeah.
I could not agree more. The wealth of this nation has depended on education, investment, infrastructure in a way that has not happened in any other country. This is a very concerning moment to watch the U.S. no longer lead the world in high school graduation and math scores, to watch other nations investing much more heavily in really advanced technologies. This is a scary moment, far more scary than some satellite going around blinking. Everything that we have invented here, the internet was amazing public investment. Google flowed from amazing public investment. The entire biotech industry flowed from amazing public investment. So to watch us right now, and the reason the president refers to this as a Sputnik moment is because I think he's trying to call attention to the tension between cutting our deficit and investing. If all we accomplish right now is cutting our deficit, but we do not invest in these things which made our nation rich, it will be a Pyrrhic victory. We will end up with a balanced budget and a second-rate nation. So how we manage to get those two things together, I don't know, but we need a conversation in Washington right now that is much more rational about what makes this nation great, and that even in the hardest times, families and certainly nations invest for the future.
It seems like a good place to pause and to say thank you to the panelists. Vicki said in her comments, now is the time. Thank you. Vicki said in her comments, now is the time to be a scientist, to invest in these discoveries, to embrace the opportunities in all you've been hearing about. Thank you so much for coming.